All right. Welcome, everyone. We are going to be starting right now. So thank you so much for coming to this Invasive Crayfish Collaborative webinar, the second of our webinar series. My name is Natalia Szklaruk. If you don't know me, I am with Illinois Indiana Sea Grant and am the new main facilitator of the Invasive Crayfish Collaborative. And for those of you who don't know what the ICC is, it's a relatively new program that brings together a variety of experts and stakeholders to address the threat of invasive crayfish. And so before we begin, I just wanted to make sure that you guys had all the resources needed to follow us and to join our membership. So if you're interested in learning more, you can follow the invasivecrayfish.org website. And that basically acts as a one-stop shop for invasive crayfish information and is always being updated. And also, if you're interested in registering for our future webinars or reviewing past recorded webinars, you can follow the last two links. And if you have any questions or concerns or comments, you can email me at szklaruk@illinois.edu. One of the goals of the ICC is to make new information regarding invasive crayfish more accessible, which in turn will make people more informed and enable them to make better, more informed decisions about invasive crayfish management. So in today's talk, we will learn about the work being done at Auburn University. So Dr. Jim Stoeckel and his graduate student Nick Barnes will discuss the recent work they have conducted regarding the development and evaluation of multiple control techniques for red swamp crayfish in surface waters and in burrows. As background, Dr. Jim Stoeckel is an associate professor in the School of Fisheries, Aquaculture, and Aquatic Sciences at Auburn University. He leads the Crustacean and Molluscan Ecology Lab, which focuses on topics such as water quality stressors, invasive species management, feeding ecology, bioenergetics, and ecotoxicology. And he holds a PhD in zoology from Miami University. Additionally, Nick Barnes is a master's student at Auburn University studying invasive red swamp crayfish under Dr. Jim Stoeckel, and his future plans include pursuing a PhD in aquatic invertebrate ecology and studying anthropologic impacts on freshwater systems. So to everyone joining us today, we encourage you to type in any questions that you might have in the Q&A box or in the chat at any point during the presentation, and then we'll eventually go through them together after the presentation ends. And also, this webinar is being recorded and it will be posted on the ICC website with captions shortly after. All right, so with that, Jim and Nick, you can go ahead and share your screen.
All right, let's see if we can get this working here. Let me redo this again. All right, everybody see that okay? Let me know if you can't. But anyway, thank you all for coming out and attending this.
Sorry, Jim, yep. I don't think I see the presentation.
I think I know what I did here. It works in rehearsal all the time, right? As soon as I think I've made a rookie mistake, and all right, now let's swap this again.
There we go. Perfect.
See that? Okay, let me have mine. All right. Well, I obviously don't know how to use them, but hopefully we'll see if I know anything more about crayfish than I do know about Zoom. But thank you all for coming out here today. What I want to talk to you today is just discuss the past probably 4 years or so of research that we've been doing at Auburn, developing and evaluating multiple control techniques for red swamp crayfish in surface waters and burrows. And here I'd like to acknowledge my co-authors because this has really been a huge group effort with lots of collaborators from different agencies. So Hisham, Gabby, Kaylin, Ben Bates, Nicole Rankin, and then of course Nick Barnes from Auburn, Aaron Cupp and Annie Alert from the USGS, and then Lucas Nathan, Kathleen, my apologies, I know you're here. I always forget how to pronounce your last name. You can see it here. And Brennan Wright from the Department of Natural Resources in Michigan. So, as we all know, Procambarus clarki, the red swamp crayfish, is native to the southeastern United States and northeastern Mexico, as we can see on this map. But it's highly invasive. It's one of the most invasive crayfish species in the world, and is highly invasive both in the U.S., as you can see by these ranges, and then the rest of the world as well. It can adapt pretty well to a wide variety of climates. And also, especially most importantly, when we're talking about control, it can adapt to environmental extremes by burrowing. And these burrows can take the form of connected burrows that have a connection to a nearby surface water, or separate burrows in the terrestrial system nearby that are not connected to those surface waters. And so what this typically means is we have to develop control methods not only for the surface waters of invaded ponds and lakes, sometimes slow-moving streams, but also for these burrows as well. Because even if you control the crayfish in the surface waters, there may be a residual population in nearby burrows that are just going to quickly repopulate those surface waters. So we've been doing this work as part of a large multi-agency research team that is funded by the Great Lakes Restoration Initiative. And this includes USGS, Michigan DNR, Michigan State University, U.S. Fish and Wildlife Service, of course, the Invasive Crayfish Collaborative, and then Auburn University. So here at Auburn, we try to develop and refine control methods in surface waters and burrows, and then evaluate the effectiveness of those control approaches using a combination of laboratory and raceway studies, as well as pond trials in semi-natural environments. And to do this, we kind of leverage the advantage of our location. So we do this work in Alabama. There is a wild population of red swamp crayfish on our research station that we can use as our subject animals. And we don't have to worry about, issues with bringing red swamp crayfish from other areas to our areas, which would be a problem, or worrying about escapement issues, because we already have a large resident population on our station to work with. So for surface water techniques, we're going to talk about a combination of pond trials and then raceway trials in the lab. And then for the burrow control techniques, we're going to talk about a combination of laboratory trials and then field trials in some of these drained ponds here. So let's start off with the pond trials in the surface waters. So one of the first things that we evaluated was using carbon dioxide as a control tool. And to do this, we utilized a series of small earthen ponds on our station. You can see that here. And at the center of each pond was a catchment basin. And we would wheel carbon dioxide tanks to the side of the pond and then bubble carbon dioxide into the center of that pond and raise the carbon dioxide level up in that water, with the hope of causing those crayfish to emerge from the pond for easy collection. We also surrounded the pond with sediment retention fencing. And the reason we did this was because we found that when the crayfish would leave the pond, a lot of times they would seek shelter underneath of this fencing. And so this was a great way to facilitate collection after they left the water. So instead of always wandering around, they would just seek shelter at one place. Incidentally, we did find that snakes really enjoyed that habitat too, which caused us a problem because we do have some venomous snakes down here that we do have to watch out for. And so during these trials, we collected both submerged crayfish that were congregating along the edges of the ponds, but were still just barely in the water, as well as crayfish that had fully emerged from the water. And so our first question was whether or not carbon dioxide could be used to induce crayfish to emerge from those ponds for easy collection. And so here you can see a picture of a crayfish that's come out and making a beeline for that shelter underneath of that netting. And so for all of these techniques, I'm just going to kind of go over the basic results and the main take-home points. If you want to know more detail, you can either ask me later, or in this case, we do have a publication that gives a lot more detail on this technique. But this graph here shows the percent of emerged crayfish on the y-axis for the ponds that we treated with CO2 versus the control ponds that received no carbon dioxide. And you can see that when we bubbled carbon dioxide into those ponds, we averaged about 12% of those crayfish would leave the pond and could be collected, as opposed to only about 1% in the control pond. So the carbon dioxide was effective, although the percentage that left the pond was not super high. It was around 12%. So we wanted to see if we could increase that capture rate. And so our next question then was whether hourly collections combined with collections of those crayfish that we would see congregating around the edge of the ponds and could be easily collected with a dip net would increase the capture rate. And so this is just some pictures of what we would see around the edges. A lot of times the crayfish wouldn't leave the pond altogether, but they'd be in that really shallow water. A lot of times they'd be on their sides, acting like they were air breathing, and they could be easily dip netted. And so again, we see the percent of crayfish captured on the y-axis, and we have the percent of submerged crayfish captured, emerged crayfish captured, and then the total number. And so if we look at the total, we can see that we did pretty good. So so when we did the hourly collections from both of these habitats, we got up to almost 50%. So about 45% of the crayfish that we put in the ponds were captured. And this was primarily due to collection of those submerged crayfish near the edge. So you can see here when we did hourly collections, we really upped our game by focusing on these crayfish that could be easily dip netted from the edges. So from this, we concluded that CO2 could be fairly effective in small ponds. Hourly collections were a lot more effective than just a single final collection, after say 5 hours of exposure to CO2. And this was primarily due to increased collection of submerged individuals near the pond edge. But this does mean that substantial effort would be required to do this in larger ponds. And so also during one of our experiments, we accidentally left on an inflow of fresh water from the middle of the pond and noticed that it really reduced our capture rate. And this is important because in some invaded habitats like retention ponds, you might have an inflow of fresh water coming into that pond, draining off of that surrounding landscape. Or in other invaded habitats, you might have an underwater spring that's supplying, or seep that's supplying fresh water to that pond. So we wanted to see whether that presence of a freshwater inflow would affect these capture rates. And so the white bars show the ponds where the inflow was absent, and the gray bars show where the inflow was present. And the answer here was definitely yes. And so when the inflow was absent, as we saw before, we got between 45 to 50% of the crayfish collected. But when there was a freshwater inflow to that pond, it went all the way down to about 20%. So it reduced it pretty substantially. So the presence of a freshwater inflow did reduce our capture rate. Fewer crayfish moved to the pond edge, and fewer crayfish emerged from that pond. We also wanted to look and see what other factors might affect the probability of capturing crayfish using carbon dioxide. And so we saw that, not surprisingly, the higher the concentration of carbon dioxide, the higher the probability that we would capture a crayfish. And so you can see here the capture probability increases pretty dramatically as you go from 0 to 400 milligrams per liter of carbon dioxide. This next graph with pH shows essentially the same thing, because as you increase the carbon dioxide, the pH obviously is going to go down in those waters. So you get the opposite relationship here. We also found that water temperature had a significant effect. So we had increased chances of capture at warmer temperatures, especially when in those hot summer days when we got up to about 30 degrees or even 35 degrees Celsius. And then crayfish size also mattered. So the larger crayfish seemed to be more affected by the CO2 and were more likely to move to the pond edge or emerge from the pond than those smaller crayfish. Because of this study, where when we had freshwater inflows, we really reduced the capture rate of crayfish, and the fact that it was pretty labor intensive to go around the edges of the pond every hour and collect crayfish, we wanted then to see whether carbon dioxide could be used to push crayfish from one end of the pond to another for easier capture. So instead of making them emerge, in which case we have to collect them all the way around the pond, we wanted to try to just push them and concentrate them all at one side of the pond for easy capture. So to do this, we used larger ponds, and we used seining material to build these large funnel traps at one end of the pond. So the seine fencing would guide them into crayfish traps that were submerged at the openings of these traps. So here you can see that we divided the pond into 4 zones, and we injected carbon dioxide into zones 1 and 2. And the hope was that we would set up a carbon dioxide gradient going from a very high concentration at zone 1 to a very low concentration at the opposite end of the pond, and try to push those crayfish all from one end of the pond to the other. And so we were successful in setting up a gradient. So this graph shows the carbon dioxide concentration in the zones through time. So we ran this over a 6-hour period, and you can see in zone 1, the carbon dioxide increased dramatically up to about, between 250 and 300 milligrams per liter. But in zone 4, it was negligible. It was maybe averaged around 10 milligrams per liter. So we got a pretty good difference there. But of course, things don't always work out the way we think they will or hope they will. And so when we looked at the number of crayfish that were captured in that low CO2 refuge area, we found that there was actually no significant difference in captures between the carbon dioxide ponds and the control ponds. So only about 4% of the crayfish were captured at that end of the pond. So we found no significant difference in crayfish captures. So that carbon dioxide was not very effective at pushing the crayfish. However, we did observe, like we did in the smaller ponds, that rather than leaving the ponds, in this larger pond, we had a lot of alligator weed growing along the edges, and those crayfish were climbing up the alligator weed and emerging to the surface of that water. And we would see this in most of our trials, but this was pretty difficult to quantify in the pond environment to actually count the number of crayfish, so they would come up and down that alligator weed over time. So we moved back to the lab to conduct some raceway trials, and so we set up these large raceways. We tried to do the same thing where we set up a carbon dioxide gradient, and then we built some emergent structures shown here for the crayfish to crawl out of the water on, and we wanted to quantitatively compare the ability of carbon dioxide to push crayfish to a refuge versus push them to emerge or not crawl out on the banks, but crawl up to a structure on the surface. So we compared push to refuge or emerge to surface. And so we had 2 treatments. The one treatment was carbon dioxide treatment. So we had freshwater inflow at one end of these troughs in zone 3, and then we bubbled carbon dioxide into zones 1 and 2, and then we set up these emergence boxes throughout these zones for the crayfish to crawl up on if they so desired. And then for the controls, we had the freshwater inflow, but no carbon dioxide input at all. And this graph just shows you that just like the ponds, we were able to successfully create a gradient where we had between 200 and 250 milligrams per liter of carbon dioxide in zones 1 and 2 here, but then very little carbon dioxide where that fresh water was coming in, where we had that freshwater inflow. We put 50 labeled crayfish in zones 1 and 2 for each trial, and then we recorded the position of the crayfish every 30 minutes. And so our results were pretty strong. So when we looked at the number of crayfish that emerged onto those structures, we found that about 20% of them would crawl out of the water and emerge onto these structures when treated with CO2. But in the control troughs, none of them emerged. They all stayed underwater. When we looked to see whether we were able to push them to the low CO2 refuge in zone 3, we again found a similar result as what we saw in the ponds, where there was actually no difference between the carbon dioxide and control treatments. But interestingly, we noticed that even though zone 3 comprised probably less than a third of that whole trough length, we got almost half of the crayfish would consistently go down into that zone 3. So they really seemed to be attracted to that zone regardless of whether there was CO2. So they also did this in the control. And so that led us to think, we weren't really thinking about flow, because crayfish, red swamp crayfish, as the name implies, are typically found in low flow, swampy areas. So we didn't think that they would be attracted to flow, but this suggested that maybe they were. And so we wanted to determine whether flow could be used to pull or attract crayfish to a specific region. So to do this, we set up another set of experiments using that same setup. But in this case, for our treatment, we had freshwater inflows in half of our troughs, but no carbon dioxide. And then in the control, we had no inflow at all in zone 3. It was just a static system. And again, we had no carbon dioxide. And so, again, we got some significant results. So this graph shows the number of crayfish that migrated to zone 3 on the y-axis. And so when we had flow in those tanks, about 37% migrated to that zone in these trials, versus in a tank with no flow, only about 13% went to that zone 3. And again, no crayfish emerged regardless of flow in the absence of CO2. So in summary, for the carbon dioxide treatments, the hourly collections of crayfish were a lot more effective than the single final collection. And we were able to get 40 to 50% of the crayfish in that pond collected within, I think it was a 7-hour period. But this was very labor intensive and would be even more of a problem in larger ponds. And this led us to try to figure out if we could use carbon dioxide to push crayfish to one end of the pond instead of emerging from the pond. But the answer to that was it does push crayfish to the pond edges and to the surface of the water, but not towards low carbon dioxide refuges. And this suggests that in future studies, we may want to try to develop and test emergence traps to be used along with carbon dioxide as an alternative to hourly collection with dip nets. So this is something we haven't tried yet, but I think might be worthwhile. We also showed some evidence that water flow might attract or pull crayfish for more efficient collection. We're currently, my student Nicole is currently finishing up some experiments to test this out, but preliminary results actually show that flow is much better at repulsing than attracting the red swamp crayfish. And so we're still working those up. I don't have enough time to go into that in detail, but I'll just give you the quick version of this set of experiments. So Nicole set up a long trough. I believe it was about 7 or 8 feet long, using gutter material, and set up 2 pit tag antennas along the length of that trough, and modulated or adjusted the signal such that the antenna could tell when a crayfish was moving. Basically, the range went out to this distance, so it could tell when the crayfish was crossing here closer to this antenna, and then the second antenna could tell when the crayfish was moving out to this zone. So using these 2 antennas, we were able to divide this raceway into 3 zones of approximately equal lengths, and we would know which zone that crayfish was in at any given time using these two pit tag antennas. We introduced flow into this raceway at one end, at the far end of the raceway, and it flowed from here down to the other end of the raceway, and then we actually introduced the crayfish at the far end of the raceway, and this set up a velocity gradient. So rather than trying to test a specific velocity, we were interested in seeing how they reacted to a gradient, because in a real world setting, if we use flow to try to attract or repulse crayfish in the field, that's what it's going to be. There's, it's going to be highest at the location of that pump or in flow, and then decrease as you get farther away. So we had a gradient going from highest to lowest velocity, and then we could track the crayfish as it moved from one end of that raceway to another. And so the one result that I'll just share with you very quickly is we looked at the proportion of time spent in zone 1 right here versus the average current velocity in zone 2. The idea being here, if this current velocity in 2 was attracting the crayfish, it would spend less time in zone 1. But if this current velocity was high enough that it was repelling that crayfish, then the crayfish would spend more time in zone 1. And so here, so that would be that velocity is repelling the crayfish. And so we tested actually 3 different size classes. I'll show you results for 2 of them. So this graph shows the proportion of time spent in zone 1 on the y-axis and average current velocity on the x-axis. And we saw a pretty strong linear relationship here. These bright green dots are the control runs where we had no flow at all. And you can see at low flow, there's a little bit of evidence for an attraction where they actually spent a little bit more time, or a little bit less time, I'm sorry, in zone 1. So they were attracted to zone 2 at very low velocities. But this is kind of weak evidence. Overall, the relationship was pretty linear, suggesting that most of the time, zone 2, those current velocities were actually repelling the crayfish, and they were spending more and more time in zone 1. So once we got above about 0.4 meters per second, they were spending almost all their time in zone 1, not venturing out. The larger size classes showed more of a threshold relationship, interestingly enough. So between velocities of 0 and about 0.4 meters per second, there was really no relationship. They spent about 20 to maybe 60% of their time in zone 1. But as soon as those velocities went above 0.4 meters per second, they spent essentially 100% of their time in zone 1. So there seems to be this 0.4 meters per second seems to be a magic number that's very good at repelling red swamp crayfish and keeping them from moving. All right, so on to the burrows. And I'm first going to talk about some field trials that we conducted to look at control techniques for burrows. And so we wanted to see whether physical blockers could effectively control burrowed crayfish. We used 4 replicate ponds. We raised and lowered the pond water levels to induce burrowing. And we tested 3 treatments per pond, 37 burrows per treatment per pond. So in our controls, we just left the burrows as they were. For treatment number 2, we used bentonite clay, and we would pour that into burrows and follow that with pouring some water down into the burrows, and that would moisten the clay and cause it to swell and form a really tight seal in that burrow. And then we also tried expanding foam that you can get from any hardware store, and you can squirt that down in the burrow. It will expand and form a plug just like the clay. And we excavated the burrows after 48 hours. And any of you who have done burrow excavation know that it can be a circus sometimes, depending on the experience of your crew. But we had a lot of fun, dug a lot of burrows, and were able to get some good results. And so this graph here shows the proportion of occupied burrows with dead crayfish in them for control, bentonite, and expanding foam treatments. And you can see here, these 2 treatments were effective relative to the control, and about somewhere between 60 to 80% of the burrows that were treated with either blocker ended up with dead crayfish in there. So it worked about 60 to 80% of the time. We wanted to know why it didn't work that other 20% of the time or so. And we found that one of the big important variables was whether or not that blocker went all the way down to the groundwater or not. So when the bentonite clay went all the way down to the groundwater, we had almost 100% mortality. And when the expanding foam went all the way down to the groundwater, we had about 80%. So that versus when it did not, our effectiveness decreased quite a bit. And we were also curious because of this as to whether it was harder to treat deeper burrows where it would be harder to get down to that groundwater than shallow burrows. But we found that that was not the case. So for bentonite clay, we found that it was actually a little bit more effective in deeper burrows. So the burrows with dead crayfish tended to be a little bit deeper than the ones with live crayfish. And for the expanding foam, there was no difference at all. So physical blockers can be effective for burrows about 60 to 80% of the time, more so if you can reach the groundwater with those blockers. So for future research, we need to develop a more efficient delivery system to get those blockers all the way down to that groundwater, and the effectiveness did not decline with deeper burrows. We would recommend bentonite clay over the expanding foam. It's less toxic because it's a natural substance. And actually, as you can see, it fills in those burrows essentially with clay. It may help to reduce damage to shorelines and levees. All right, so now I'm going to turn the talk over to Dick, my student, and he's going to talk about some lab trials that he conducted to test some techniques for crayfish and burrows.
All right, thank you so much, Jim. So I'm going to be going over the studies I've been doing for the past year and a half, looking at different factors that contribute to the burrowing behaviors of red swamp crayfish, and then looking at using hot water as a potential control method. So first, going over the reasons that red swamp crayfish burrow, mainly are coming down to these three reasons, being predator evasion, reproduction, and seeking water. Like Jim mentioned earlier, they are native to areas that are very drought prone. So being able to follow the water line down gives them the ability to stay in areas that many other species are going to get evicted from when the water is going away. So we were studying burrowing in two different forms. On the left, this is a picture taken in the Atchafalaya Basin in Louisiana, where this species is native to. Down there, they are often forming more shallow burrows with multiple entrances and chimneys. This picture in the middle, that is a research pond at Auburn. These are 40 by 40 clay line ponds with a catch basin in the middle that allow us to raise and lower water levels based off of study needs. And then finally, on the far right are the ant farms or the burrowing chambers. And these give us even more control over burrowing studies and also give us the ability to see what the crayfish are doing while underground. So the first study we ran with these burrowing chambers was looking at how do different drainage rates affect the burrowing behavior. So to do this, we ran 9 burrowing chambers with individual sumps and foraging areas 4 different times. In between each run, each burrowing chamber was taken down and disassembled with new water and burrowing substrate added to reduce the possibility that any pheromones left behind by previous crayfish could impact any burrowing. The animals we were using for this species were large red male red swamp crayfish. These were connected less than 100 feet from the station. Like Jim said, we have a natural population, so no problem to get those. And previous studies have shown a difference in the burrowing behaviors between males and females, so to keep it constant, we just used the males. The burrowing soil was a high percentage clay sieved through a 1 centimeter mesh. This clay is a common pond liner found in Alabama and is able to support more intricate burrow structures. And additionally, other studies have shown a preference towards clay soil over sandy or loamy soils. And then finally, the conditions that these studies were run were to replicate spring environmental conditions in Alabama, which is when we start to see burrows form. So that was a 24 degrees Celsius air and water temperature with a photo period of 12 hours on, 12 hours off. So a little bit more about these burrowing chambers. They are a 2x4 frame with pieces of transparent plexiglass sandwiched in between. And then in the middle of this, there is a vinyl gutter frame. And this vinyl gutter frame is what keeps the burrowing soil separate from the outer water channel. The water is pumped from the sumps down below, fed up and down, and then will feed out through a standpipe here on the left. These gutter frames have holes drilled in on either side, and then on the inner side, there is mesh glued in. So that allows water to flow in and permeate through into the burrowing arena while keeping that soil stuck right there. And then on top, this is where the animals were introduced. These are the foraging tanks, and on the bottom are the individual sumps. So our 3 treatments that we ran was for 12 of them, we dropped the water level for 4 centimeters a day for 5 days. The middle group was 8 centimeters a day for 3 days. And then finally was 24 centimeters all at once on day 1. And just to note, prior to crayfish introduction, the burrowing chambers were totally saturated with water all the way up to the top for at least 24 hours. And then it wasn't until the crayfish was introduced to the foraging tank that we started to drop the water. So the results that we see from this study, for the first group, the 4 centimeter a day for 5 days, of those 12 crayfish, we had 11 burrow every day. You see in this picture on the left, this crayfish constructed a burrow and followed the water line down all 5 days to this 24 centimeter mark. For the middle group, the 8 centimeters a day for 3 days, of those 12 crayfish, we had only 7 burrowed. However, none of these crayfish burrowed after the first day. You see here on this middle picture, this crayfish followed the water line down to day 1, that 8 centimeter mark, constructed a burrow, but did not follow the water line down for days 2 and 3. And finally, for the static 24 centimeter a day group, of those 12 crayfish, we had 0 burrow. In fact, we had 3 of them desiccate out on top, even though the water was only 24 centimeters below. So, to start to develop more control methods and looking at how effective hot water is as a control method, we need to know what the thermal limit of red swamp crayfish is. Additionally, since this species has such a large global distribution, we also need to know, does acclimation temperature affect red swamp crayfish response to hot water? So to do this, we set up 2 different troughs, one held at 15 degrees Celsius, one held at 25 degrees Celsius. And within these, we housed 80 crayfish, 40 males and 40 females for at least 7 days to provide acclimation to those acclimation temperatures. And then for the actual hot water studies, we set up hot water baths in coolers using independent thermometers and heat probes. And these were set up in between 36 degrees Celsius and 46 degrees Celsius in increments of 2 degrees. We submerged glass jars, and the crayfish were placed in those for 9 minutes, and every 3 minutes we looked at 2 different mortality endpoints. The first of which was a riding response, and a loss of which would indicate functional death, and last being response to probing, and a loss of that would indicate physiological death. So to do this, you can start to see the difference in the groups. For the lower 36 degrees Celsius group, those crayfish are still very active and resistant to the riding response flip. That holds true for the 38 degrees Celsius group. They are still able to right themselves very quickly. However, we start to see in the 40 degrees Celsius group lethargy, while being able to still right themselves within 30 seconds. For the 42 degrees Celsius group, while these still respond to probing and aren't physiologically dead, they have lost their ability to right themselves. And then finally, for the 44 degrees Celsius group, these crayfish all died before the 3-minute mortality check. And the main difference we found between the 2 acclimation group, for the lower 15 degrees Celsius, we found 100% loss of riding response after 3 minutes exposure to 38 degrees Celsius, and 100% mortality after only 3 minutes exposure to 44 degrees Celsius. But for the warmer 25 degrees Celsius group, we had 100% loss of riding response after 3 minutes exposure to 38 degrees Celsius as well, but it took 6 minutes exposure to 44 degrees Celsius before we reached 100% mortality. So now that we know how these crayfish are going to start to respond to hot water, we can start to do field studies to evaluate the effectiveness of it. And then, as we saw in the burrowing chamber studies, this species prefers a slower draining soil. So to do this, we set up soaker hoses along the upper perimeters of our research ponds to continue to provide moisture, to hopefully provide a constantly moist environment to hopefully promote them burrowing during the hot Alabama summer. To get the hot water into the burrows, first we started with an off-the-shelf commercial crayfish cooking kit. However, with this, we were only safely able to boil 10 gallons of water to 70 degrees Celsius, which is neither hot enough or enough water to treat a single burrow. Next, we went with a more industrialized heated pressure washer. However, we found with these, they require either a constant water line or a gas line, which is not something we can provide in all of our field sites. So finally, we custom ordered a 2-stage pressure washer with a diesel burning burner and a gas-powered pump with its own independent 250-gallon water tank on a skid frame. So this gives us the ability to load it in the back of a pickup truck and take it into the field where there is no water or gas pickups required. So using this pressure washer, we defined a treatment as the total time spent spraying 100 degrees Celsius water into a burrow after reaching 70 degrees Celsius. We split these treatments into 3 different time intervals, first of which being 30 seconds, next being 2.5 minutes, and finally 5 minutes. We chose these time intervals thinking about what land managers might experience in the field. If they could treat 100 burrows at 30 seconds apiece, this might be an effective treatment strategy. However, if we're still seeing low mortality after 5 minutes of treatment per burrow, we know that we might need to move on to a different strategy. And just a reminder that in the lab studies, we were seeing 100% mortality after only 3 minutes exposure to 44 degrees Celsius. So you would think that spraying 100 degrees Celsius water into burrow is going to have very, very high mortality. However, when we ran these studies on 45 burrows, we found that we were getting around a 60 to 67% mortality regardless of the treatment. So we plotted the mortality against the burrow depth on graphs here. And so for the 30-second treatment of those 15 burrows treated, we had 10 with dead crayfish and found that there was around a 70% probability of death if the burrow was less than 80 centimeters. For the 2.5-minute group, of the 15 burrows treated, we found only 9 with all dead crayfish and found an 85% probability of death if the burrow was less than 60 centimeters. And then finally, for the 5-minute group of those 15 burrows treated, we found 10 with dead crayfish and found around a 90% probability of death if the burrow was less than 60 centimeters. And so finally, just a couple of confounding variables that we will start to address when we analyze this data even farther is the amount of burrow or water in a burrow. Some burrows, when you open the plug or get to them, they have no water and it takes a while for the water to start to fill out. And some have water standing at the surface. And we found the burrows with water already standing at the surface, the crayfish was most likely a little higher in the burrow and we were able to flush it out. But the burrows that had less water, those crayfish were able to get down into their chamber with the mud and stick down in there. And then additionally, a total number of entrances. Not only does this apply to different species, but also this species in different geographic regions. Like we saw in Louisiana, this red swamp species will dig burrows with 2 to 3 entrances. But in Alabama, we are seeing mostly just single entrance burrows. And what that can do is not only provide an escape for that crayfish to maybe get away from the hot water, but also potentially provide thermal venting to get some of the heat out of the burrow. And then next, I will hand it back over to Jim for him to finish out.
All right, awesome. Thanks a lot, Nick. Appreciate that. So I just have 2 more slides. I just want to briefly touch on a couple of other projects. The first is at Auburn and also in Michigan, various parts of our group have conducted crayfish telemetry studies where we glue acoustic tags to the back of the crayfish. This allows us to get the location of crayfish within sub-meter accuracy, so very precise location. And I think we could get locations maybe every 10 seconds or so, or every minute. Actually, I think it was closer to every 10 seconds. And so we could really look at the response to different treatments. And so this figure here just shows the example of a 10-minute crayfish track in a pond, and you can see we were able to get very high resolution movement paths of those crayfish. And then this is kind of zooming out, showing the tracks of multiple crayfish in one of our experimental ponds over multiple days. And this information, or these experiments, are going to be described in more detail in upcoming seminars by the USGS and or the Michigan crew. So stay tuned for details on this. These were pretty cool studies. And then also, I just wanted to put in a plug for an upcoming project we have where we are going to be investigating the use of neofemales to control invasive red swamp crayfish. And this was a technique that was proposed for control of red swamp crayfish by Sabaya et al. In 2020. And the way this works is that crayfish males are genetically ZZ. So unlike us, the males are homozygous. And they have a gland at the base of their fifth legs called the androgenic gland. If that gland is removed or inactivated at a very early age, that genetic male, instead of developing into a reproductive male, will actually develop by default into a reproductive female. So this is called a neo-female. So even though it's a genetic male, ZZ, it will produce ovaries and eggs and be able to reproduce. And so if you cross a neo-female, that's ZZ, with a normal ZZ male, that ZZ by ZZ, almost sounds like a rock and roll band. That ZZ and ZZ cross will actually produce all ZZ male offspring. And so the idea here is that we might be able to use this technique to depress the reproductive potential of an invasive species. And so this project will be divided into 4 phases. The first phase is we're just starting now, and that involves conducting a literature review of the state of the science, establishing a collaborative science team of experts, and then identifying the expertise and facilities needed. In phase 2, we will begin production of neo-females by developing and testing neo-female production protocols, and then develop population models to estimate the numbers needed. Phase 3, we hope to do some proof of concept pond trials where we test model predictions using experimental ponds. And at this point, we'll be initiating the necessary regulatory processes for field trials. And then in phase 4, we hope to conduct field trials in invaded ponds. At this point, before we do this, we'll be completing any regulatory requirements and non-target risk assessments, and then hopefully apply to invaded ponds in areas such as those in Southeast Michigan. And since we've got a good crayfish group here, let me just put in a further plug for this, in that we do have funding to hire a postdoc for this project, and we're advertising now. So if any of you know of any good postdocs that might be interested in this project, have them contact me. And with that, I'd like to thank you all for sticking with me and Nick as we go through this whirlwind of projects. I'd like to acknowledge our funding source. This was funded by the U.S. EPA's Great Lakes Restoration Initiative under Focus Area 2, using a cooperative agreement between Auburn University and the USGS. And then I'd like to just thank a long list of people that provided assistance in the field and in the laboratory. And if you have any questions that we're not able to answer in the next few minutes, here, my email and Nick's email are listed here. And so we'd be glad to answer any further questions if you send us emails. And with that, I thank you all, and I think we're ready to open it up for questions.
Great. Thank you so much, Jim and Nick. That was fascinating work. Thank you again. So again, we encourage anyone to write any questions. Currently we got a question, sorry. How deep was the hot water injected?
So that really depends on burrow morphology. Some burrows will go down like a foot and then do a 90-degree bend. Some do more like perp through actions, and then some do just go straight down 3 or 4 feet. So the ones that were more straight on, I would get the wand as deep as possible, but sometimes just based off the burrow design, you could only get it in a couple inches.
I'll follow that up with a plug for our friends in Montana. So there's a group in Montana that actually is a hatchery manager up there that actually came up with this idea. And they actually were able to attach a flexible hose to their applicator. And we just met with them yesterday, and they were able to actually, with that flexibility, get it pretty deep into the burrow. So they were able to spray water, navigate those turns, spray water pretty deep into the burrow, and they feel like they've had comparable success to what we had as well. So we're hoping to use that as something. Of course, once you hear about it, it's like I don't know why we didn't think of that to begin with, but we will try to use that in the future, try to get that hot water injected a little bit deeper from the beginning.
Great, thank you. I actually had a question about the CO2 concentrations that you used in those pond trials. I was wondering what those concentrations were.
Yeah, so I'll answer that while I'm zooming back through here. But essentially, we would try, our target concentration was 200 milligrams per liter, and that was based on some laboratory trials that had been conducted previous to this. And if you can see here, this kind of answers your question here, I think, this graph. So this is the probability of capture based on CO2 concentration. So so this was our target, but sometimes we were able to get it all the way up to 400 milligrams per liter. And that really increased the probability of us getting crayfish.
Did you observe any mortality with those higher concentrations of CO2?
No, and that's a great question. We did not observe crayfish mortality, although sometimes the ones along the edge, they weren't fully sedated, but they might be moving a little bit slower. But one of our collaborators with USGS, Aaron Cupp, has been doing some lethal carbon dioxide assays, and he will probably be talking about that a bit when he gives his seminar. So stay tuned for that. But not at the levels we tested. We did not see high mortality of crayfish.
Oh, did you see mortality in other
Yeah,
organisms?
so we didn't have anything other than crayfish in our ponds. But again, this may come up in a future seminar. When this technique was applied in Michigan, the fish in some of those invaded retention ponds didn't do as well as the crayfish, and so there was some mortality with some of the fish in those ponds, I believe. But again, I mean, I know that they observed them floating to the surface. I wasn't there when they did that trial, but that'll be a good question for the USGS crew.
Got it. Let's see. Any other questions? I guess I had another one, if that's all right, while other people are possibly coming up with their own questions. Out of the different control techniques that you guys analyze, the CO2 and the velocity strength and the physical blockers and hot water, which one do you think is the most effective at controlling invasive red swamp crayfish? Or maybe is it a good idea to use all of them at once?
Yeah, I think the answer is it depends. And so that's kind of the reason we're doing these trials and trying to figure out the strengths and weaknesses of each one. So, a lot depends on the conditions, on what your goal is. And I think there's no one single silver bullet. So, our probably conclusion is that for effective control, you're going to have to utilize a number of techniques in combination with each other and probably apply them repeatedly. And so our goal here is try to just figure out what's the effectiveness under different conditions. So, for CO2, for example, these kind of graphs can help you know that, if your temperature is really cold, might not be a good time to try to use CO2. If it's warmer, might be a better time. The hot water seems to be, if it pans out, it seems to be as effective as the physical blockers. Both of those are pretty good because they're, well, the clay, not the great stuff, but the clay and then the hot water would require no permits. And because the hot water is pretty comparable to the clay, very few people are going to object to you filling a burrow with hot water. So you don't have to worry about all the regulatory issues. But if you also want to try to seal those burrows and repair any damage to a levee, probably be better off with the clay, because even though, Nick has been asked before, what's the pressure of that water going in? We try to do a low pressure water going into the burrows so that we're not damaging those burrows and just washing things out more. But, at best, that water is not going to repair the damage. At worst, it might cause a little bit more erosion if you spray that in too hard. So again, it kind of depends on your location and what your goals are and what the environmental conditions are.
Yeah, that makes sense. We got another question. How much time elapsed between injecting the physical blockers and excavation? Do you think if the
days.
burrows were left longer? Sorry, there were two questions here. I guess you
Okay.
could answer that first question. How much time elapsed?
Yeah, so 48 hours. So we dug them up in 2 days.
And then the second question along with that was, do you think if the burrows were left longer,
Okay.
mortality would be higher as they used up the oxygen?
Yeah, that's
Yeah.
a really good question. So I think so. The reason for that is we never saw crayfish excavate a side burrow. So one of the worries was that the crayfish were going to be able to kind of dig another burrow around that blocker, and if they did that, they'd be getting air right back down in there. We never did see evidence of that. We would put uh, some fencing around each burrow so that if the crayfish did come out or did dig another side burrow, we'd be able to capture it and see that. We never saw that, and so yeah, presumably that oxygen is going to run out over time. We also do work in my lab where we look at the ability of crayfish to withstand anoxia. Red swamp crayfish are pretty good at it, and they can survive 0 milligrams of oxygen per liter. I'd have to look at the paper to get the exact number, but I think it was around, I think they averaged around 7 to 10 hours. And that's no oxygen at all. And so in those burrows, if that blocker doesn't get all the way down to the water, there's going to be a little bit of airspace. So it's just gradually, going to be hypoxic and take a while to go down to anoxia. So that's a long answer to, yeah, I think if we left those over multiple days, we might have seen higher mortality. And that's a great point.
Great. Thank you so much. So that is all the time that we have today. Oh, we got another comment. If you're researching ways to control the red crayfish, do other crayfish share burrows? That's a good question, actually.
Yeah, so that's a big concern. So other crayfish don't share the burrows with the red swamp crayfish, but if you're going to be controlling burrowing crayfish in an area, you really need to know what other burrowing crayfish are around in your area. It's actually pretty difficult to tell species from the crayfish burrows. So just by looking at a burrow, it's tough to definitively say, is this a red swamp crayfish burrow versus something else? Sometimes you can do it, but you have to have a very experienced person. So that's a concern. You want to make sure that the majority of your burrows that you're treating are in an invaded area that's dominated or is just exclusively red swamp crayfish. There are other organisms that do share crayfish burrows occasionally. Some of those are of conservation concern. So the Hines emerald dragonfly in some regions, which is an endangered dragonfly species, does live in crayfish burrows during the larval stage. And so that would be a concern in areas with them. There is a species of rattlesnake, which I can't remember the name offhand, that's of conservation concern. Luckily, it's not around here, or I would never stick my hand down a crayfish burrow. But there are some parts of the country within that range where they do get into crayfish burrows. And so again, when you're treating burrows, you need to do some homework. And like you said, make sure there's no other burrowing crayfish of conservation concern around, as well as other organisms that might share that burrow.
Great. Thank you so much. And we'll end it on one last question. We got one more in the chat. For those that lived, how often did you see crayfish dig back out of the bent seal?
We did not.
Versus the burrow was still sealed? I'm sorry.
Yes.
Yeah.
Yeah. So we never, during those experiments, we never saw a crayfish dig around that bent seal plug. And then Nick, I think Annie said yesterday, they've tried this technique. They use bent seal up in Michigan now for invaded ponds. And again, this will be something to ask during the USPS seminar, but I'm pretty sure that they told us yesterday. Kathleen's on this call, so she might have a better answer than me, but I'm pretty sure that they do not see very few, if any, exit holes around the bend seal when they treat up there in Michigan as well. So it's possible, but it doesn't seem to happen very often.
Great. Okay. Thank you so much. Oh, is someone?
Yeah, this is Kathleen. I just wanted to pipe up. I was actually my question because we do see crayfish come back up through the bend seal.
Okay.
They don't dig a separate hole, but if the bend seal doesn't get hard enough, they'll just come back through the bend seal, and you'll see like a little. So I was wondering if you ever saw that.
No, we did not. But remember that we only looked at these burrows over a 2-day period. So do you see that after 2 days, Kathleen?
I'm not sure how long it usually is because we are hitting so many burrows. We're not usually checking them every day. But it'll be within a week probably for sure. You'll see like a little hole in the bend seal where they've crawled back out. And that I think is usually either because it was too wet or it wasn't filled enough.
Yeah, so if you have that, it gets down to that air. If you have that airspace for them to work with, that gives them some room to excavate. And so, yeah, it'd be interesting to follow that up and see if it's the same thing. If you get that bend seal all the way down to the burrow water, to the bottom of the burrow, are they able to do that? So that's good to know. And I'll just say here again, this is something, always take the lab studies and the experimental pond studies with a grain of salt, because a lot of times things may work really well in the lab or in experimental systems, but then when you apply it in the real world, there's a lot of other factors come into play, and it may or may not work as well as what we see in these controlled experiments.
Thanks, Kathleen. Thank you, Jim. Okay, so that's truly all the time now that we have today. I want to thank you guys again, Jim and Nick, for taking the time and coming out and talking to us about your work. And thanks again to everyone who attended this meeting. Just as a reminder, this recording will be posted on the ICC website with captions. And then we'll be actually having another webinar held in November. I believe 2 of them in November, but more info will come out on the events page soon. So again, thank you so much for being here. And I hope everyone has a great rest of their day. Thank you so much.
Thank you.